Chapter number 1. Biotechnology. Scope and importance. What is biotechnology? In 1917, a Hungarian engineer, Karl Erecki, coined the term biotechnology to describe a process toward large-scale production of pig. According to him, all types of work are biotechnology, by which products are produced from raw materials using living organisms. During the end of 20th century, biotechnology emerged as a new discipline of biology integrating with technology, but the root of biotechnology lies in biology. There was no sudden sprout of this discipline, but some of the methods for production of products were developed centuries back. Therefore, biotechnology is concerned with exploitation of biological components for production of useful products. Biotechnology is defined by different organizations in different ways. It has been broadly defined as the development and utilization of biological processes, forms and systems for obtaining maximum benefits to man and other forms of life. Biotechnology is the science of applied biological process. Biotechnology, a Dutch perspective, 1981. Following are some of the definitions given by other organizations. Biotechnology is the application of scientific and engineering principles to the processing of materials by biological agents to provide goods and service. The Organization for Economic Cooperation and Development, OECD, 1981. The integrated use of biochemistry, microbiology and engineering sciences in order to achieve technological application of the capabilities of microorganisms, cultured tissue, cells, and parts thereof. The European Federation of Biotechnology, EFB, 1981. O'Sullivan, 19811. The application of biochemistry, biology, microbiology and chemical engineering to industrial process and products and on environment. International Union of Pure and Applied Chemistry, IAPAC, 1981. Biotechnology is the controlled use of biological agents such as microorganisms or cellular components for beneficial use. U.S. National Science Federation. In the definition given by OECD, scientific and engineering principles refer to microbiology, genetics, biochemistry, etc. And biological agents means microorganisms, enzymes, plant and animal cells. The meaning of these three definitions and others given by many organizations are more or less similar. A unified definition of genetic engineering has been given by Smith, 1996, as the formation of new combinations of heritable material by the insertion of nucleic acid molecules products by whatever means outside the cell, into any virus, bacterial plasmid or other vector system so as to allow their incorporation into a host organism in which they do not naturally occur and which they are capable of continued propagation. B. History of biotechnology. If we trace the origin of biotechnology, it is as old as human civilization. Development of biotechnology can be studied considering its growth that occurred in two phases, I. The traditional, old biotechnology, and E. The new, modern biotechnology. 1. Traditional biotechnology. Really, the traditional biotechnology is the kitchen technology developed by our ancestors using the fermenting bacteria. Kitchen technology is as old as human civilization. During Vedic period 5000, 7000 BC, Aryans had been performing daily agnihotra or yajna. One of the materials used in yajna is animal fat, i.e. ghee, which is a fermented product of milk. Similarly, the divine soma, a fermented microbial product used as beverage, had been offered to God. Sumerians and Babylonians, 6000 BC, were drinking the beer. Egyptians were baking leavened bread by 4000 BC. Preparation of curd, ghee, wine, beer, vinegar, etc. was the kitchen technology. In spite of all development, preparation of curd, ghee, vinegar, alcoholic beverages, jalebi, idli, dosa, have become an art of the kitchen of all Indians. For more detail see Table I. The traditional biotechnology refers to the conventional technology which have been used for many centuries. Beer, wine, cheese and many foods have been produced using traditional biotechnology. Thus, the traditional biotechnology includes the process that are based on the natural capabilities of microorganisms. The traditional biotechnology has established a huge and expanding world market. In monetary term, it represents a major part of all biotechnology financial profits. In India, who can forget the story of Makhan Chori, butter stealing, by Lord Sri Krishna during Mahabharat period? Butter would have been produced following the same kitchen art. Besides breeding of strong productive animals, selection of desirable seeds enhanced crop production has been the part of human activity since the time immemorial. The Egyptians, about BC, used to prepare vinegar from crushed dates by keeping longer time. But the crushed dates produce intoxicants at first. In Egypt, Mesopotamia and Palestine, about BC, the production of wine from crushed grapes, and beer from germinated cereals malt, using a bread leaven, a mass of yeast, was established. In Indian Ayurveda, production of asava and arista, using deterrent substrates, and flowers of mahua, maduka indica, or dataki, wadfordia fructicosa, has been well characterized till today. Since Vedic period, in these methods various substrates are transformed into a number of products. Hence, the odor, color and taste of final products are changed. Moreover, use of salt for preservation of various food has been earlier developed even in Europe. Still, preservation methodology of the mummies of Egypt are noteworthy. Possibly, mummification would have been done through dehydration of dead body followed by use of mixture of salts, largely sodium carbonate. Thus, the traditional biotechnology was an art rather than a science. Role of microorganisms in fermentation. The causes of fermentation could be discovered after observing microorganisms using a microscope by Antony van Leeuwenhoek, 1673, 1723, at Delft, Holland. During 18th century a significant contribution was made by the chemists on the process and products of fermentation. In 1757, it was demonstrated that a milky precipitate could be obtained when the gas evolved from fermentation was passed through the lime water. It was called lime water test. A similar gas comes out from burning of charcoal. Henry Cavendish demonstrated that the gas evolved from brown sugar in water treated with yeast was absorbed by sodium hydroxide solution. Shale and J. Priestley, 1772, 1774 confirmed the identity of oxygen gas present in air. Using analytical technique for carbon estimation, Antoine Lavoisier gave the chemical basis of alcoholic fermentation. A French man, Nicolas Appert, 1810, described the method of food preservation. In the same year Peter Durand also gave the use of tin container for food preservation. It was done by putting an airtight vessel containing food material in the boiling water. It increased the importance of canning industry. Lack of oxygen in such a closed and heated vessel was reported by Gay-Lussac. He concluded that oxygen was required, or initiation of alcoholic fermentation, but not for further progress of fermentation. After 183, Charles B. Astier gave the concept that air is the carrier of kinds of germs. In 1837, Theodore Schwann, after a series of experimentation, demonstrated that the development of the fungus, sugar fungus, on fruit juice causes fermentation. He was the first to observe and describe the yeast in growing process. Charles Conyard Latour, 1838, observed yeast budding using a microscope allowing 300 to 400 power magnification. Justus von Liebig, 1839, a well-known chemist, proclaimed that all the activity of yeast cells was the result of chemical and physical reactions going on in the medium. The study of microbiology was started since the first report of Louis Pasteur, 1857, on lactic acid fermentation from sugar. He isolated the microorganisms, lactic yeast, that were associated with lactic acid and formed curd. The cells of lactic yeast were smaller than that of beer yeast. Lactic acid production got increased when he added chalk powder to fermentation medium. Pasteur showed the presence of lactic acid in curd by using a polarimeter. In 1860, Pasteur provided a detailed report on the use of synthetic medium for microbiologic studies. He concluded that fermentation is carried out anaerobically by the living cells. The yeast increased the weight with increase in C and N contents of overall batch during alcoholic fermentation in synthetic medium. The increase in yeast protein in synthetic medium was accompanied by a related decrease in the ammonium nitrogen in the medium. Fermentation of sugar was required for multiplication of yeast cells. Similar phenomenon occurred in fermentation of lactic acid, tartaric acid, butyric acid. Because of not using pure culture, some of the fermentation were stopped. Growth and physiology of yeast differ when they are grown under aerobic and anaerobic conditions. It was later on called as Pasteur effect. Under anaerobic conditions a large amount of sugar was converted into alcohol, while under aerobic conditions large amount of sugar was converted into yeast cell mass. Pasteur suggested that high percentage of microbial population is killed by heating the juice at 62.80 C 1450 F, which is now called as pasteurization. Robert Koch, 1881, gave the method for establishing relationship of a pathogen with a disease, which is known as Koch's postulates or pathogenicity test. Following this technique, he proved that the anthrax disease is caused by Bacillus anthracis. Edward Buchner, 1897, was the first to demonstrate enzymatically mediated fermentation reactions. He showed that cell tree yeast juice mixed with concentrated sugar solution evolved upon incubation the carbon dioxide and produced ethyl alcohol. Similar products were also produced in aqueous solution of the other sugars such as glucose, sucrose, fructose and maltose. Buchner called the dissolved substance in juice responsible for sugar fermentation as zymase. This work showed the improved techniques of fermentation. Fermentation could also be demonstrated in test tubes without a living organism. The work of chemistry had more role in understanding the fermentation phenomenon than the microbiology. During 1890s, Hans Buchner and Martin Hahn developed more effective method of getting cell-free extracts after disrupting the microbial cells. They ground the microbial cells with quartz and adding kieselguhr to get sufficient consistency in the resulting paste. Thus, sucrose fermentation using cell-free extracts from yeast cells, obtained by the above method, was demonstrated. Discovery of viruses and their role in disease was possible when Charles Chamberlain, 1884, constructed a porcelain bacterial filter. Edward Jenner, 1798, used vaccination by taking out liquid material from cowpox lesions and introducing into people having smallpox. But Pasteur and Chamberlain developed an attenuated anthrax vaccine against anthrax disease. After the discovery of toxins produced by Corynebacterium diphtheriae causes diphtheria in human, its antitoxin was developed by Emil von Behring, 1890, and Shibasaburo Kitasato. He injected the inactivated toxin into rabbit that induced to produce antitoxin in the blood. The antitoxin inactivated the toxin and protected against the disease. Similarly, tetanus antitoxin was developed by Behring. 2. Modern biotechnology. The two major features of technology differentiates the modern biotechnology from the classical biotechnology. Capability of science to change the genetic material for getting new products for specific requirement through recombinant DNA technology, and ownership of technology and its sociopolitical impact. Now the conventional industries, pharmaceutical industries, agro-industries, etc., are focusing their attention to produce biotechnology-based products. Emergence of modern biotechnology. The new or modern biotechnology embraces all methods of genetic modification by recombinant DNA and cell fusion technologies. It also includes the modern developments of traditional biotechnological processes. The new aspects of biotechnology founded in recent advancement of modern biology, genetic engineering and fermentation process technology are now increasingly find wide industrial application. But the rate of application will depend on adequate investment by the industries, improved system of biological patenting, marketing skill, economics of new methods, and public perception about the biotechnology products. With the end of 19th century, the traditional biotechnology associated with fermentation was gradually industrialized. This resulted in gradual growth of industries producing beer, whiskey, wine, rum, canned food, etc. In 1920, for the first time, the Leeds City Council, UK, established the Institute of Biotechnology. In the late 1960s, OECD was set up to promote policies for sound economic growth of the member countries. In 1978, the European Federation of Biotechnology was set up. In 20th century, biotechnology brought industries and agriculture together. During World War I, fermentation processes were developed which produced the acetone from starch and paint solvent from automobile industries. During World War XI, the antibiotic penicillin was discovered. Manufacture of penicillin shifted the biotechnological focus towards pharmaceuticals. Linking the fermentation with biochemistry, bioprocess, chemical engineering and instrument designing helped substantially in the progress of industries. During Gulf War 1991, the work on microorganisms dominated for the preparation of biological warfare, antibiotics and fermentation process. Suspected preparation of biological and chemical warfare led to U.S. attack on Iraq in 2003. After the discovery of double helix DNA by Watson and Crick 1953, Werner Arber 1971 discovered a special enzyme in bacteria which he called the restriction enzymes. These enzymes can cut the DNA strand and generate fragments. The cut ends of two fragments are single-stranded sticky ends because the single-stranded ends having identical base pairs can rejoin. In 1973, S. Cohen and H. Boyer removed a specific gene from a bacterium and inserted into another bacterium using restriction enzymes. This discovery marked the start of recombinant DNA technology or genetic engineering. In 1976, Baltimore successfully transferred human growth hormone gene into a rabbit. In 1978, the European Federation of Biotechnology was created. In 1978, a U.S. company Genentech used genetic engineering technique to produce human insulin in E. coli. In 1980, trials of new hormone was conducted in the USA, France, Japan, and the United Kingdom. The U.S. Food and Drug Administration gave marketing approval to Humulin, i.e. human insulin made by Eli Lilly, USA, by the end of 1982. Another hormone, somatotropin, was produced on industrial scale. In 1993, the first genetically engineered tomatoes, Flavorsaver, were sold in market. In 1996, the first clone lamb Dolly was born successfully by the efforts of scientists of Scotland. Thereafter, several cloned animals were produced. In 2001, the sequence of the human genome was published in Nato and Science. Human Genome Project was completed by March 2003. In 2002, a designer baby was born to cure the genetic disease of her elder sister. On December 27, 2002, a claim was made for the birth of a clone baby Eve, by the scientists of Human Cloning Society, the Clonate of France. In February 2005, in the meeting of United Nations, many countries opposed gene cloning in humans, while a few countries supported with request to allow for the sake of only. In May 2005, scientists in South Korea have used a method called therapeutic cloning to produce stem cell lines. These are genetic matches to patients. Such stem cell lines could be used for disease research. The USA condemned this approach. In this method, human embryos were produced through cloning, as done for Dolly, and stem cells were obtained from blastocyst. The excised stem cells could be grown in vitro and used further. It is, therefore, interesting that the scientists are engaged in doing the most challenging task of mass production of growth hormones, insulin, vaccines, immunogenic proteins and polypeptides, gene therapy, biofertilizers, biopesticides, producing disease and stress-resistant plants, biomass, enzymes, antibiotics, acids, fuels, etc. Many biochemical companies such as National Pituitary Agency, USA, Lilly, USA, Kabi Vitram AB, Sweden, Genentech Company, USA, Biogen, Switzerland, etc. are trying to produce some of the above products by using genetic engineering techniques. Many Nobel laureates including Dr. Har Govind Khurana, are associated with these companies. The area of main interest, at present, where such works are being done throughout the world are given in Table 1.2. Many international agencies are making efforts to solve the national and international problems via collaborative works. Various programs including molecular and environmental engineering have been taken up by the European communities with framework of their collaborative activities. For training purposes, under several biotechnological programs, centers have been established, for example, the International Center for Cooperative Research and Training in Engineering, Japan, and Institute of Biotechnological Studies. Many countries have developed collaborative networks on several aspects such as Regional Microbiology Network for Southeast Asia, supported by Japan and UNESCO. Microbiological Resource Centers, MICRCENS, supported by UNESCO, United Nations Environmental Programs, UNEP, International Cell Research Organization, ICRO. These two organizations have participations of groups in many countries. See Table 1.2. Areas of biotechnology. B. Biotechnology is an interdisciplinary area. Biotechnology is not a sudden discovery, rather a coming of age of a technology that was initiated several decades ago. By the middle of 20th century there had been a tremendous growth in the area of chemistry, physics and biology. Further knowledge of each branch have been advanced substantially. Multidisciplinary strategies were made for the solution of various problems. A novel spectrum of investigation occurred through the true interdisciplinary synthesis. This led to the evolution of biotechnology, which is an outcome of integrated effort of biology with technology, the root of which lies in biological science. Figure 1.1. The key difference between biology and biotechnology is their scale of operation. Usually the biologist works in the range of nanograms to milligrams. Biotechnologists working on the production of vaccine may be satisfied with milligram yields, but many other projects aims at kilograms or tons. Thus, the main objective of biotechnologists consists of scaling up the biological processes. Smith, 1996. The main discipline of biology are microbiology, biochemistry, genetics, molecular biology, immunology, cell and tissue culture. However, on the engineering side it includes chemical and biochemical engineering such as large-scale cultivation of microbes and cells, their upstream and downstream process, etc. These processes can be separated into 5 major operations: I. Strain selection and improvement, E. Mass culture, E. Optimization of cell responses, IV. Process operation, and V. Downstream processing, i.e. product recovery. Many areas of biotechnology have arisen through the interaction between various parts of biology and engineering: biochemistry, biophysics, cell biology, colloid chemistry, embryology, ecology, genetics, immunology, molecular biology, medical chemistry, pharmacology, polymer chemistry, thermochemistry and virology. The modern biotechnology has developed technologies extracting the basic knowledge from biology. See Figure 1.2, Biotechnology Tree. C. Global impact and current of biotechnology. Each and every organism performs its function within its optimum limits. The excitement about the modern biotechnology is that the scientific methods, such as genetic engineering, have enhanced the natural capabilities of natural production of organisms. What a miracle is that a mouse of the size of rabbit can be produced. Bacteria like E. coli are producing mammalian hormones such as insulin, somatostatin, somatotropin, etc. Yeast cells have been genetically manipulated to produce vaccine against hepatitis B virus, hepatitis disease. Myeloma cells, cancerous cells of bone marrow, and B cells of immunized mice were hybridized to produce hybrid cells that consisted the characteristics of both the cells, which were cell division and antibody production. Now the hybrid cells, hybridomas, are being used for production of monoclonal antibodies. In 1982, interferons A, cis and gamma were produced by genetically engineered E. coli cells. Techniques have been developed to produce rare and medicinally valuable molecules, to change hereditary traits of plants and animals, to diagnose diseases, to produce useful chemicals and to clean up and restore the environment. In this way biotechnology has great impact in the fields of health, food, agriculture and environmental protection. Due to rapid development, the present situation is that there is no difference between pharmaceutical firms and biotechnology industry. However, approved products in the pipeline and renewed public confidence made it one of the most promising areas of economic growth in future. India offers a huge market for the products as well as cheap manufacturing base for export. Pod, 1996. Following are some of the areas where biotechnology has done the best. I. Health care. The maximum benefits of biotechnology has been utilized by health care. Biotechnology-derived proteins and polypeptides form the new class of potential drugs. For example, insulin was primarily extracted from slaughter animals. Since 1982, human insulin, humulin, has been produced by microorganisms in fermenters. Similarly, hepatitis B vaccines viz. Recombavax Fibs, from Merck, Guni at, from Shantha Biotechnics Limited, Hyderabad, Shanvac at Biological E Laboratory, etc. are the genetically engineered vaccines produced biotechnologically. Since 1987, the number of biotechnology-derived new protein drugs has surpassed the new chemical drugs. Table 1.3 shows some of the important products produced through genetically modified organisms. With the advancement of gene manipulation in organisms, the science have led to a new revolution in biology which is called gene revolution. Obviously, it is a third revolution in the science after Industrial Revolution and Computer Revolution. Thus the roots of today's biotechnology lies in chemistry, physics and biology. See Table 1.3. There are about 35 biotechnology-derived therapeutics and vaccines approved by the US FDA alone for medical use, and more than 500 drugs and vaccines to reach in market. Table 1.4. Similarly, about 600 biotechnology diagnostics are worldwide available in clinical practices. Table 1.5. About 130 gene therapy protocols have been approved by the US authorities. India relies on imports of many immunodiagnostic kits. See Table 1.4 and 1.5. Agriculture. Biotechnology is making new ground in the food agriculture area. Current public debate about BSTC, bovine somatotropin, a hormone administered to cows to increase milk production, typifies an example of biotechnology product testing public acceptance. Similarly, the flavorsavertum tomato, produced by transgenic plants engineered by antisense technology to preserve flavor, texture and quality, is a new breed of value-added foods. Food biotechnology offers valuable and viable alternative to food problems, and a solution to nutritionally influenced diseases such as diabetes, hypertension, cancer, heart diseases, arthritis, etc. A transgenic golden rice has been produced by introducing 3 genes for the production of vitamin A in type A rice. Several insect-resistant transgenic Bt plants have been produced by incorporating insecticidal toxin-producing Bt gene of Bacillus thuringiensis into the desired plants. A transgenic cotton named Ingard was released in Australia which contained Bt gene and provided resistance against insects. We can recall the Bt cotton prepared for Andhra Pradesh but sown in Gujarat in 2001 which raised debate throughout the country. Biopesticides are coming to the market and their sales are increasing. Molecular farming is a new concept where therapeutic drugs are produced in farm animals, for example, therapeutic proteins secreted in goat milk. There are about a dozen companies that produce lactoferrin, TPA, hemoglobin, melanin and interleukins in cows, goat and pigs. However, it is not surprising that vegetables producing vaccines, insulin, interferon and growth hormones would be available in market in 21st century, beside human clones and several other miracles. Human Genome Project (IIGP). The major landmark in human history is the Human Genome Sequence. The HGP is an international research program. Almost the whole human genome has been sequenced and chromosome map has been developed in various laboratories worldwide through coordinated efforts. Human chromosome mapping was completed by March 2003. There are 33,000 functional genes in human. More than 97% genes are non-functional. They do not encode any polypeptide chain. Objectives of Human Genome Project are to construct the detailed genetic and physical map of human genome, determine the complete nucleotide sequence of human DNA, store information in database, locate the estimated genes within the human genome, address the ethical, legal and social issues that may arise from the project, perform similar analysis on the genomes of several other organisms. Environment. The natural biodegradability of pollutants present in environment increased with the use of biotechnology. The bioremediation technologies have been found successful to combat the pollution problems. See Chapter 25, Environmental Biotechnology. Bioremediation is the use of microorganisms to detoxify pollutants, present in the environment usually as soil or water sediments. The pollutants cause several health problems. Microorganisms which show potential to degradation of oil, pesticides and fertilizers belong to the genera of bacteria Pseudomonas, Micrococcus, Bacillus, and fungi Candida, Cladosporium, Torolipsis, Trichoderma, etc. Genomics and Proteomics. Computer-based study and designing of genome is called genomics. Genomics deals with sequencing of the complete genome of a particular organism. Similarly, study of proteins present on genome using computer is called proteomics. The proteomics can be defined as the study of all the proteins present in genome of an organism. With the help of proteomics and genomics, the new molecules that can interact with the other partners could identified. This gives us deep insight into biological pathway. Now the whole genome is available to biologists for scrutiny. There may be new small molecules as potential drug candidates. Therefore, interaction of some molecules can be studied in greater detail. The 33,000 genes of human are on a microchip. It has helped to design drugs for genetic diseases, for which there is no cure so far. For example, a specific gene, HER2/neu, overexpresses in breast cancer patients. A designed drug, Herceptin, is good for treatment of breast cancer. Thus, the field of genomics has helped the growth of pharmacologic, toxicologic and protein studies on animals. Therefore, the new areas are called pharmacogenomics, toxicogenomic and proteogenomic, respectively, see Chapter 8. Bioinformatics. It is a new field of biotechnology linked with information technology. Bioinformatics may be defined as application of information sciences, mathematics, statistics, and computer sciences to increase the understanding of biology, biochemistry and biological data.